Hello, my name is Aaron Omel, and I am recording this teaching demo for the Odasis SI position. Specifically, I'll be teaching general biology. Now, if I am chosen for this SI position, my first step in my class would be to basically put the name of the organizer that we're doing on top of the board before the students get in. And now for this demo, I'll be going over the world history topic. Now, when the students first walk in, I'll be going over the whole of the organizer, and after I go over the organizer, I will ask the students I'll be giving them questions to do, and having them write their own organizers. Now, for world history, I would ask them what the first step is, what the first step in world history is, and the first step would be RNA world. The RNA world hypothesis, specifically. And also in that same step is the iron sulfur hypothesis. Now, right around here, I will go more in depth into what these two topics mean. Now, the RNA world is basically the hypothesis that life started from these little things called the ribozymes that can self-replicate but aren't exactly alive. And the iron sulfur theory came about from the theory that life came about from hydrothermal vents at the bottom of the ocean that spewed iron and sulfur into the water that eventually sedimented on the bottom, giving way for life to gain these resources and become more complex. Now, in this same step, I'll also go into how you can relate these steps to other topics. Like example for here, RNA world, what I've drawn here is a protocell. And this protocell is also in the abiogenesis topic. The reason why I do this is to have the students remember past topics and not just go over world history, but also go over abiogenesis as well and other topics. Now, going back to world history, the next step is prokaryotes. Prokaryotes. Around here, I'll also be asking the students why. Why specifically is prokaryotes why specifically is prokaryotes the next step instead of what the next step is. This will help the students further memorize what a prokaryote is and also why it is the next step in the process. It is the next step because it is the simplest life form, or the next simplest life form, because they don't have nucleus, they don't have membrane-bound organelles, and it is the simplest life form at this step. Now, prokaryotes also have other forms at this stage called autotrophs. Now, autotrophs are the next step because in order to be alive, you have to gain energy from the environment. And the only way to get energy from the environment is to be an autotroph. And this consists of the sun, getting energy from the sun, or from carbon cycling, and other sources of energy. Now here, heterotrophs, they do not make their own energy. So they have to consume other organisms, autotrophs, to get their own energy. Now, the next step is the cyanobacteria. Now, around here, I will ask the students what major event has the cyanobacteria contributed to? And if they can't answer, I'll answer for them. And the answer is the O2 apocalypse. And on top of this, after I've given them the answer, I'll ask them why. Why is the O2 apocalypse important at all? And it is important is because it destroyed all organisms that could not process O2 because it was toxic to them. They all died out, and the only remaining organisms were oxygen intaking organisms. Now, after this, we have the theory of endosymbiosis. Now, around here, I'll ask them to make their own organizer of endosymbiosis to see if they truly know what endosymbiosis is, since it is a more complex topic than what a prokaryote is. And after this, I'll go over what endosymbiosis is, and if they have questions, I'll answer them, and I'll fill them in if they have any wrong parts on their own organizers. Now, endosymbiosis is basically the creation of eukaryotes. And how this happens is a heterotroph decides to eat an autotroph. But instead of digesting the autotroph, they form an endosymbiotic bond. And in doing this, they create a eukaryote that has both its own cell membrane and another one inside of it. Now, I can link this to another topic as well, which is the, where I can ask them another question on how to prove that the theory of endosymbiosis really happened. Ask them why why we know the theory of endosymbiosis has weight to it. And the reason for this is because the mitochondria has its very own DNA. This proves that the mitochondria is its own organism because it has its own DNA and its own way of reproducing. Now around here, the eukaryote line splits. And it splits because the cells eat different organisms. For example, this cell on the left has eaten what will soon become a mitochondria. And this has produced the animal line. Every cell that has eaten a mitochondria will all become animals. But over here, we have a cell that has eaten a mitochondria and a chloroplast. Now here, all of these cells will become plants. Very, very long down the line. And around here, I'll ask the students why this is. Why do all the organisms that have consumed a mitochondria and a chloroplast become plants instead of becoming animals? This is because, as you can see right now, only plants have chloroplasts. No animal at all has a chloroplast. That's how we know that all of these have become animals in some sort of way and all of these have become plants in some sort of way. And after I've gone over this organizer with students, I will ask them to create their own organizer on the next topic that we'll be going over. And after they have done that, I will go over their own organizers to see if they have anything wrong on them. For example, if I ask them to make an abiogenesis topic right over here, I will tell them first what they should do, the 4 steps, and ask them to fill out what they are. And after waiting for them to finish, I will go over it with them, and if they have any questions, then I'll go in more in depth with them. Now, the first step for abiogenesis is the introduction of abiogenesis, or the formation of abiogenesis. Now, while I'm going over this, I'll also ask them what are also the 4 requirements for abiogenesis. right? So right around here, we link this back to the cyanobacteria apocalypse, which is that organisms cannot process O2 at this time, so they need low oxygen, low O2. And bring it back to here, the for abiogenesis to happen, they need energy, free energy. And they also need lots of time to progress themselves. And they also need the basic elements to survive, which is a little acronym that I created for it, which is CHON: carbon, hydrogen, oxygen, and nitrogen. After doing this, I'll go over the next step, which is the formation of micromolecules, organic micromolecules. Around here, I'll ask them why. Why is organic micromolecules important to these steps for abiogenesis? And I'll answer it because it is important because they need organic macromolecules to further gain more energy and become more complex in the future. Now, the next step will be protocells, the formation of protocells. This goes back to what I said here about RNA world, basically stating that protocells, the organism needs an phospholipid bilayer. And I can also link this to phospholipid bilayer. I can link this back to the fluid mosaic model, while also going over this topic. And lastly, for the steps for abiogenesis, we have introduction of self-replication. Right? So while I'm going over with the students, I'll branch off into multiple different categories, basically helping them to remember multiple different categories while also going over world history, our own topic. And in the future, I'll go further back to older categories to make sure that my students learn and retain the information that I'm teaching them. And at the end of my class, I will give them a homework assignment based on what I have taught them today. For example, I'll ask them to basically write down what are the 4 steps of abiogenesis or some other branching points that they can, or I'll also ask them to make other branching points for themselves based on the topic we went over today. Other branching points that they can come over for themselves and further reach out and help them create their own organizers so that they can make one of these for themselves when they study by themselves. All right, so this concludes my teaching demo for the Odasis bio position, and I hope you consider me for the job.